We welcome Linden Fay.
Can everybody hear me? Okay. Hello, everybody. My name is Linden Fay. I am a rising junior majoring in computer science and mathematics. And before I begin, I would like to thank Hofstra and Feinstein for the opportunity to participate inside the Asbury program. So this summer, I got to join Dr. Eric Chang and the rest of his research team at Feinstein. The main goal of our project is to basically analyze how neurons develop, communicate, and respond to different conditions, as well as study how they interact in different environments. This is the primary research focus of our lab at Feinstein. And some key terms that we're going over, we need to analyze how they work together in vitro, which essentially means outside of the living organism and inside a dish, as well as in different co-culture settings, which means how they interact with different cell types together. This here is a picture of a tree. What may be an unassuming picture at first actually holds some key integral details regarding the structure of neurons in vitro. Think about the trunk of the tree as the core of the neuron, and all of the branches and the sub-branches as different neurites, all with their different paths. This is the overarching structure of the neurons that we're trying to analyze in this project. So onto what my task was. I used Python coding to essentially automate this process of neuronal structure analysis. What the program is supposed to do is basically figure out all the structure that includes finding out where the core is, where all the branches is, and be able to quantify that in certain parameters. So why is this important? So these morphological changes in neuronal shapes tell a lot about many of its functions, how it relates to disease, its viability and health, and many other functions about the neuron. Some of the measurements that we'll be taking with this program include each of the neurites' length and widths, the amount of branches each neurite has, and the number of endpoints and branch points on the neurite. So over long video recordings of live cells, there is going to be thousands of individual frames to process manually. Essentially, how this task is being approached right now is purely through manual tracing. We're basically just tracing out the center, drawing all the paths of the branches, and this will take a very long time given all these different time frames of neuronal images. That is the goal of this program, to basically automate this process and basically speed up how we can analyze the parameters of its structure. So a quick overview of how this program works. Here is one of the pictures of neurons that we have analyzed in the lab. What we did here is we marked the neuron with TD tomato, that is the red color that we see, and then we placed it under an echo confocal laser microscope that allows us to use fluorescent imaging to pick up on the red tint and better visualize where the neuron is. From here, we can take this image and plug it into our pipeline. So the first step is to pre-process this image. What we do here, we need to grayscale the image. We need to brighten up the contrast. That allows the software to detect the neuron better. And we also threshold it to make it really clear where exactly the neuron is going to be in this image. Next, we isolate the soma and skeletonize the dendritic structure. So we can see the soma outlined here through this green outline around the center of the neuron. And the dendritic structure is essentially where all the branches, all the neurites are going to be. That's what we see here with the red outline. We can also see sometimes in certain images, due to brightness difference, there can be some gaps inside of these branches that we need to close shut. So what we do here, we need to stitch these gaps together. What we do here, we basically use an algorithm that detects these gaps in the branches, and then we stitch them shut to make sure that our neurite branches are able to pathfind all the way from the center of the neuron all the way to their outmost endpoints. Next, what we do is we need to define what exactly these neurites are. So a neurite, what defined is basically starting from its center node all the way to their outmost points. That is how we pathfind the neurites in each of these neurons. So how we do that, we use the soma we detected earlier, the center of the neuron. we find the center nodes of the neuron, which is these small red nodes here. What these essentially are is basically their neurites up to their first branch point, which is up unto their first point of divergence. We can see from here, it goes into separate paths. And then we find these separate paths traced directly back to the center with these pink outline. So what this does is it lets us define what the neurite paths are, and it also clears up the noise we had on the corners before because they aren't traced directly back to the center of the neuron. Lastly, we need to export this data and visualize it in plots and so forth. We figure out all of the structural parameters we get from these neurites. We can get their center node coordinates, we can get their lengths, how they relate to each other. We put them in Excel and we can better visualize them in plots and graphs later on. So I would like to present two test cases that basically show how this program is applied to analyze the structure of these neurons. In the first case, we analyze one of these neurons, how it grows over certain time frames, over 60 hours, this neuron. So what we do here is we have certain frames of this neuron at different time frames. We can already see that it slowly grows its neurite branches over time. On the last one, we can see the left branch here has definitely grown a lot more compared to the previous images. So once we put it in the program, we can basically outline where that skeleton is. The program detects all of its neurite branches and basically outlines it in red. Then we need to define some other structural parameters we need to get from this red skeleton. In the first image here, we have all of its endpoints and branch points. So the red points, the endpoints, are every single time we reach the end of a path. So there's only one way we can go from that specific area. And the blue points, the branch points, are anytime we have multiple diverging paths that we can go. So like for example, here, there are multiple paths we can go from this area. So from here, we use this information and we're able to basically put it into different graphs. So here we can analyze the longest neurite length over time. We can clearly see that has a very drastic increase throughout the length. Basically, we can see from here that slowly the length grows until we have this huge growth spurt on the rightmost image of the time frames. As for the total branch length over time, it also follows a similar trend of increasing. There is a small dip on the second time frame, and that's the neuron over here. This is probably because we can see here that the skeleton has kind of detached itself from the rest of the neuron. It's not directly connected to the center, and is therefore not tallied in the total length of the branches. This is probably an issue in the brightness detection of the program, which we plan to resolve at a later date. On to the second case. Here we analyze different neurons in different co-culture settings. So on the left images, we have neurons with colonic epithelial cells. And on the right images, we pair neurons with MC38 cancer cells. Here we can see how the different neurons grow over time and how their environment greatly impacts how their health and how their growth is over the time of their lifespan. We can see here that in all of these graphs, it is very clear that MC38 cancer cells co-cultured with neurons definitely show a much larger growth spurt in terms of their branch length, number of branches, total neurite length, and the longest Euclidean distance from each of their centered nodes outward to their furthermost points of the neuron branches. So in conclusion, we have successfully created a program in Python to automate this process of neuronal structure analysis from fluorescence microscopy images. Basically, we have analyzed all these images taken under our echoconfocal microscope, and we're basically able to analyze them with this program, pick out all of their structural parameters and quantify them in data and with plots and graphs. So some next steps for this program include improving processing quality with image segmentation, which could also help alleviate some of the brightness issues we're having on some of the images, as well as adapt the program to better recognize and analyze multiple neurons in the same image. This will also save lots of time because currently what we're doing is channeling all the neurons, separating them in advance before we plug them into the program to analyze them one by one. So being able to do that innately in the program will also definitely be much more efficient in terms of analyzing their structure. In the future, this tool can also be used to quantify some other aspects of neurite growth, as well as also how they register static inputs, certain signal processing parameters, and other key factors in cancer neuroscience and analyzing them in other neuron and tumor cell co-culture interactions. Are there any questions? Yes.
Linden, great presentation. I'm curious, have you noticed any kind of fractal dimension where you can tell how long a neuron usually become in length before it starts branching? And then when it branches, did you also look at the diameter of the neurons to see how does that daughter branch compare in length and diameter compared to the mother branch?
So right now what we're doing is we're tracing all the Neurite paths. So any branch points we have will be recorded as separate branches as part of that Neurite. In terms of diameter, we are still working on that. Right now what the skeleton does is it reduces all of these branches to 1 pixel size, which allows us to more efficiently calculate all their lengths and so on and so forth. We have tried diameter, and so far the methods we've used for that kind of make the skeleton have multiple branches on the same Neurite, as in like the same 1 pixel size will have multiple paths on that same branch. So we're trying to find a way to overcome that as well.
So if you take your program and apply to a picture of the tree you showed first, which has a really robust branching pattern, would your program be able to predict like the fractal dimension used in nature for a tree, how often it branches, at what length scale, how the daughter branches, like 1.4, I'm sorry, it's about 40% of the diameter of the original or so forth? Can you use a test case like that with your program to see if it's able to provide those answers?
To an extent, yes. What the program does is it basically uses contour detection to find the center. So hopefully it should detect the trunk of the tree as where the core of it is. And then from there, it finds all the skeleton, which is all the branches of the tree, and basically outlines it from there. One thing to consider is that there's going to be lots of overlapping points, right? So like the tree, the picture we showed before has a lot of points because it's 3D and not 2D under a disk, right? So we have to find a way to account for all the parts where it overlaps. One way we're doing that right now is basically finding all the endpoints and then finding the shortest path to that endpoint. That makes sure that there are no repeated branches and everything is calculated at least once. So that's one way we can kind of quantify the structure of other things, like apart from neurons as well.
Thank you. One more.
Have you validated the results that you've gotten with a manual calculation of the branching lengths to ensure that it's the correct measurements?
Yes, we have done some of that. Essentially, what we just do is that like what we've been doing already previously, which is just tracing it out. So anytime you can take one of these images, trace it out, that takes around like 10, 20 minutes for every single one of these images. And we can basically just compare like that to see how the results differ.
Okay, thank you.
Thank you.
Thank you, Linden.